human body is a very complex system. And within that, many more layers of complexities are added when the hundreds and thousands of microbes that inhabit the gut and the mouth and the skin all play a role. Scientists at TCS Research are trying to analyze how microbes influence the human body and how the human body plays a role in the entire genome or the microbiome or the microbe bouquet which lives within us. All being done to try and understand if signals can be found on how this microbial community can give diagnostic markers. And scientists at TCS Research are trying to decipher many of the unknowns, all in an effort to give us personalized medicine in the future. Complexity simplified. I am inside one of India's illustrious companies, TCS, Tata Consultancy Services, the pioneer in IT for India. IT and TCS go together. I am here not to talk about software or IT, but to talk about life sciences. Yes, TCS also does life sciences, and to explain to us how TCS is helping keeping India healthy, we have with us Dr. Sharmila Mande. Namaskar.
Namaskar.
Dr. Sharmila is the distinguished chief scientist at TCS Research, and she has spent a long time looking at different genomes. Dr. Sharmila, TCS and life sciences, how do they fit in?
So, TCS, we do a lot of research. The work on the life sciences started, the research part started more than 20 years ago. When I joined TCS in 2001 to start the life sciences R&D group. And over time, we have started, realizing the importance of the genomics and there are different, biological problems which we have been working on. So that's why
Now, your key area of work is looking at genomes. Most of us have heard about human genomes, but you look at microbial genomes, correct?
Yes, that's absolutely right. When people talk about genomes, they understand our genome because we are made up of DNA. We have a genome of corresponding to that. But there are several microbes. Each of them, they have their own DNA, and to understand those DNA of those microbial communities are very, very important. And over the last 15 years or 20 years or so, people have realized, they play a very important role in our health and well-being. They are present everywhere. They are present inside us. They are present outside. If you talk about, soil, water, they are everywhere. But we have been focusing on looking into the genomes of these microbial communities, which are present within us.
Is it correct that in my human body, there would be microbes would outnumber my cells, and that would be true for all humans?
Yes, absolutely. So it has been estimated that we have almost equal number of microbial cells, or maybe more. But what is important is that although they are equal number, the number of genes which they have is many more. Like for example, we have 25,000 genes in the human genome. But if you consider the complete microbial population which is there in our body, they are almost like 3 million genes which they have. So they play a very, very important role in our health.
To understand how humans and microbes interact and how the microbes inside me help in my survival, let's now go out into the open air, into a park and understand that. Let's go, ma'am.
Thank you.
We are now in the sylvan surroundings of the TCS offices, and there are microbes everywhere, in the soil, in the air, in the water we drink, in the food we have, and inside me. And we are talking about microbes which live so much inside us. Dr. Sharmila, we are living in COVID-19 conditions and people have so much fear of microbes and viruses. But microbes and humans, is it a win-win situation?
You know, when people talk about, we think that, microbes are all bad because we know the viruses cause diseases, there are bacterial infections and so on. But in reality, there are lots of microbes which are present. You just mentioned the soil, air, water, and within us, they are actually useful. So they help in doing several functions which, our own genes cannot do. So for digesting food, for example, right? So we eat different kinds of food, but can we digest? Can our DNA digest all that by itself? No. There are lots of them, lots of the bacteria which are there, lots of microbes which are there which help in assimilating those foods and then getting absorbed in the body. So they are as important as for our health.
So humans and microbes which live in me is a win-win situation?
Yes, absolutely, they are. Without microbes, I don't think we will be able to survive the way we do. So we definitely need microbes, the sets of microbes which we need for.
Now, how do we, when children are born, do they already have this set of microbes in their gut? Or how do we acquire them?
So, what scientists have shown that when the babies are born, depending on the mode of delivery, whether it is a normal delivery or a cesarean, they get an inoculum of microbial community. If it is normal, they get from their mothers. If it is from the cesarean, they get from the person who was or the nurse handling the baby. So they get the inoculum right at the birth, and as they grow, they start stabilizing, depending on the way they eat and drink and so on. So first few years, 4 to 5 years, is very crucial for getting a stable microbiome, healthy microbial community, or what is called as the microbiome. I just said the word microbiome, which is the community of microbes which reside within us or around us. And within a couple of years, they get stabilized in the body. And most of them are in the gut, as you rightly said.
Is it any other organs or it's mostly in the gut? I believe it's more than a kg of microbes in our body.
So, what these microbes exist in all the organs which are exposed to the external environment. For example, our skin.
Oh, skin will also have microbes?
Skin has, definitely. So we have done work on the skin microbiome. So the community existing on the skin. So they are in the skin, oral cavity, gastrointestinal tract, in the lung, because we breathe in. So they are present in all the places on the body sites which are exposed to the environment.
And all of these live like commensals. So they live there, benefit from us, and also benefit us.
Yes. So there are all these microbes which are there. There is a nice harmony between the community which is there and within them, between our cells and those microbial cells. there's a nice balance. But if there is an imbalance which happens, particularly in the microbial community, then we get into problem.
That's what they say, that I have a gut feeling something is going wrong.
So I think I completely believe that, when we say gut feel, it's not the feeling of the gut, this is the feeling of the microbes.
So the microbes have pre-warned or prejudged that something is going wrong.
So they probably sense the signals in some way, because this community of microbes, they do some function. So it's not just they are there. Like, the community of microbes which you see around us, right? There is they are doing certain functions. They are doing some job, right? So they have some skills which they use those skills. Similarly, the microbes also have some skills and which they utilize either for digesting the food or sensing something which is external agent which comes in, and so on. So the function is very, very important for these microbes.
Dr. Sharmila, does the microbiome which is there inside you and me, does it change over time, and does eating habits and geography also affect the microbiome?
Yes, you asked a very pertinent question. All these microbial community which are there, as I said, that they start at the birth, they get that, and then over time they stabilize. But once they stabilize, depending on the lifestyle, the food we eat, and the way we work, the geography, ethnicity, all these things, they matter, because they decide what kind of community of microbes will be there, or the microbiome. So each one of us are very unique in that. Almost like 2/3 of our microbes which are there might be unique. But if we compare ours, microbiome with the Western world, there will be very different, because the food habits are very different. But the food what we eat here are very, very different. So depending on those, it depends on which microbes like those food and utilize them in what way, and so on.
And occasionally, can these bacteria turn hostile to us, or these microbiome turn hostile to the human body?
So this microbiome, what happens is that there is a fine balance amongst all the community of microbes which are there in the body. But at times, the balance might get lost. The imbalance, and that's when all the problems start.
But who decides this balance? The biome, the microbes, or the humans?
So it's actually it's a very different question. Who is driving? It's our genome, because it's our genome or these microbes. But I think it's a combination of both. It's actually a intertwined one. So our genome decides on the microbes, what kind of microbial community. Of course, the other factors like environmental factors like diet, ethnicity, all those factors come into picture. But also, in these microbes also influence the way we behave, and so on. That has been actually shown.
And do these microbes or the microbiome affect only the gut, or does it go to other organs which are far away from the gut?
So so normally you would think that if you have microbes in the gut, it will affect, and if there is an imbalance, it will affect only the gut. But people have shown that the imbalance in the gut microbial community, right? So that affects not just the health of the gastrointestinal tract, but also other organs like behavior, heart diseases, the lung functioning, muscle functioning. All these are also been linked with what kind of microbial communities are there in the gut.
And how do you do your sampling? How do you get these microbiomes from humans to your own facilities where you test them?
So if I have to look into, for example, our gut microbiome, right? So the gut microbial communities which are there in the gut. So what we do is just take a spatula full of stool, and which actually gives a representation of from there. If you can analyze and see that what kind of microbes are there in your large intestine, and that's where the majority of the bacteria are there. So we basically take these stool samples and then get all the DNA of all the microbes which are there in that particular stool sample, and then lot of experiments which go on.
Let's now go into the sequencing laboratory to understand how Dr. Sharmila deciphers the mystery of the genome of the microbes and how it interacts with the genomes of humans. Let's go back. We are now inside the wet laboratory of TCS Research, where sample processing happens and the sequencing of the metagenomes. Dr. Sharmila, how do you get the samples from the field, and what is this processing which happens, and what is she doing?
So what we do is that the samples we collect from, say for example, if it's the gut microbiome which you want to study, you just simply get the stool samples, just the spatulas full of stool, or if you want to see the oral cavity, you take the saliva sample. And what we have here is, this is the sample, one of the samples which we have collected in this, and this, what she is doing is she is then processing those, because what is there in this particular solution, what is here, they are all the different microbes which are present there. And what she is doing, that she is trying to extract all the DNA out of those, DNA of all the microbes which are present there, and those DNA are then finally going to be sequenced. So these samples are stored in here later on. She is going to extract all the sequences, and then we do the molecular biology to do further processing to see the quality of the DNA what is extracted.
Let's go and see the other aspects of sample processing. Dr. Sharmila, what happens here and what is the next step?
So as I said, that we first make sure that pre-process those samples so that you get the right kind of fragments from these DNAs of all the microbes which are there. So what she is doing is, she is actually doing the molecular biology experiments to take out the required DNA areas of the DNA, which can be put it into the sequencing, which is the next step.
And then you do the sequencing? Then
we go for sequencing.
Dr. Sharmila, is this the heart of what you do? Is this the sequencer?
So this is the sequencer, where we get the samples which I mentioned, that we prepare the samples, and the sample comes in, it's loaded in this. There are about 96 samples can be done. This is a high throughput sequencing machine, and where the sequences, the samples are loaded, and then what we get are the DNA sequences of all those microbes which are there, their fragments, they all come out from these sequencing machine.
Now, one part, how does some of this work we have seen get to understanding gut health?
So you have rightly pointed out that the gut health is definitely very important, because what has been seen is that whatever microbes are there in the gut, they are linked to diseases and disorders which affect different body parts, including our brain, heart, lungs, and so on. So it's important to understand gut health. So what we have done is that we have developed an algorithm looking at just not the microbes which are present in the disease and compared it with the healthy, but what we have done is we have identified these microbes, which are about 16 microbes, which together can be considered as marker, because we have looked into the functional aspect of this microbe, not just the presence, because when you look into a community, they are doing certain functions. So in a healthy gut, the functions of these one subset of these microbes will decide whether the health of the gut is good or not. So in a disease, we have actually checked it out with the publicly available data sets of the healthy and the disease cases, and we did find that the gut health of the healthy individuals are indeed much higher when we compare with our, the gut feel metric, what we have developed in the lab.
Dr. Sharmila, let's go out and talk about the bigger picture and understand how you're doing work on diseases. Having understood how microbial genomes and human genomes and their metagenomics interact, let us try and understand how all of this then plays into healthy lifestyles, and can this information be used to decipher and do early detection of diseases. Dr. Sharmila, we've seen how you sequence the genomes of microbes. Ultimately, how do they play? Does the metagenomics and the microbiome get affected by disease, and is a diseased person's microbiome and a healthy person's microbiome different?
You're absolutely right. When we look into the microbiome from the disease and the healthy and they compare the two, what you see is the microbial community structure, which I have highlighted earlier also, that they are very different in the disease cases as compared to in the healthy cases. There are certain microbes which are very prominent in one, whereas in others which are very different. So they are very, very distinct, and people have been working on this, including us. We have been working on understanding the difference between the healthy and the diseased person, looking at different diseases, and they do differ.
Now, you've also looked at the microbial genomes between malnourished children and healthy children, and that is one area of work where you excel. Tell me a little more about that.
So this was my very first project which I did along with our collaborator from National Institute of Cholera and Infectious Diseases. We looked into the microbiome from children from rural area, and which had varying nutritional status. So we looked into this particular spectrum and tried to understand, is there any link between malnourishment and the kind of microbes which are present in their guts. And indeed we did find that there are certain microbes which are actually the community of microbes that differ as you go from severely malnourished or a healthy to a severely malnourished child. So the community structure keeps changing. And in the malnourished, what we see is that the bad ones, the pathogen ones actually dominate, those communities dominate. Whereas in the healthy, although there are the pathogens, they're not able to do anything. So it's like they can't bully on the good ones.
So you also worked on preterm births and how that can be used, the microbiome can be used as a marker, a diagnostic marker for women who may be predisposed for preterm births. Tell me a little bit about that.
So this work, what we had done was we actually was trying to understand, is there something which is different in the preterm cases as compared to the term cases, particularly the microbiome in these women, the pregnant women. And we came up with the idea that we realized that it is important not to just look at the dominant microbes which are there in any, whether if you take a disease case or a healthy case, in this case a preterm versus a term, what was realized that it is important that there are other microbes which are there, which are not in large amount, which are not the dominant ones, but they are also doing some important function. You know, they are doing they have some important role which they have. So what we are trying to do, what we have done in that particular study is that we have identified, we came up with a metric called as the taxonomic composition skew, which actually captures the dominant as well as the sparse microbial groups, and which can predict the risk of preterm delivery from the pregnant woman as early as in the first trimester, or in the first trimester we can predict with more than 95% accuracy.
95% accuracy?
Yes.
And you can save babies if you are able to get it accurate.
Correct. So actually if you are talking about saving
lives,
babies, there are 15 million babies who are born preterm, and about 1 to 1.5 million babies, they die every year because of the preterm complications. So just imagine if we can predict the risk of preterm right in the first trimester, then doctors will give preventive measures so that the instead of preterm, they will go almost to the term, or if not in the term cases, and it will save lot of these deaths of this baby.
These microbes tell whether Pallav will get some cancer earlier.
In fact, I mean, we have been looking into different diseases and disorders, and if the preterm birth, prediction risk was the first one which we started with. Then we realized why not look into all other diseases, and then we started looking into cancers, autism, schizophrenia, and so on. And what we have is we also have a different kind of algorithm which we developed, looking into the functional potential of the microbes, which can predict the risk of different cancers, colorectal cancer, breast cancer. These are the ones which we have, and this is just simple stool sample. We are looking into the gut microbial, or just the saliva sample. So it's all non-invasive. So you can predict very early on, and once it is predicted that you have a risk, then it can be suggested that the doctors can go for further tests to confirm that, or look into other tests which they would like to go for.
Dr. Sharmila, if you have understood some of the markers, are these tests available? I can go and get myself tested, or when will they be available?
So whatever I said is really the future, what it is going to be. It has to go through the validation which is going on for some of those biomarkers which we have for the risk prediction.
So will their work at some point lead us to personalized medicine?
Yes, I think the future is going to be the personalized and preventive and personalized healthcare. As I said, that there is an imbalance which happens in our microbiome, because of which leads to diseases and disorders. So one can think of personalized diet or the personalized therapy which can be recommended, and that's something which we are actually working on. My group has been working on understanding which kind of food supplements will help taking care of that imbalance, so that it comes to the balanced state, including for example in the malnourished children.
Thanks a lot, Dr. Sharmila, for explaining the balance, or the fine balance between microbes and human body, and how it affects our life, lifestyle, and the future of personalized medicine, and all available just by some diagnostic markers which you analyze through the metagenomics. Thanks a lot for speaking.
Thank you so much. Thank you so much.
There are microbes all around us, and humans and microbes have evolved together. We are living currently under COVID-19 conditions, where a single virus has given us a lot of trauma. But microbes and humans, they have happily lived together and evolved. Now scientists at TCS are trying to decipher some of the mysteries of how the genomes of humans and the microbes interact. A futuristic, personalized medicine and a delivery mechanism towards that. Keep watching India Science.